Diffusion, the international science radio show.
We have a bouncer on the doors of perception.
The good, the bad, the ugly.
It gets pretty exciting.
The myths, the truths.
Toxicology.
Astroseismology.
Magnetism. The dark side.
Genetically engineered potatoes.
Planetoid.
I
love that word.
Hello and welcome to Diffusion. Sit back and relax while we network your brain with weird and wonderful science. I'm Ian Woolf. On this edition, I bring you the 2024 Nobel Prizes in Physiology or Medicine, Physics and Chemistry. It shouldn't be too big a surprise that AI dominates the field. Genetic networks. The Physiology Nobel Prize for 2024 went jointly to Victor Ambros and Gary Ruvkun for the discovery of micro-RNA that regulate the production of proteins by genes in cells, a little like a communications network between different genes. Their work helped explain how, despite all our cells carrying the same DNA, they can produce different proteins and have different characteristics. For example, nerve cells and muscle cells are highly specialized for different functions, but all the cells have the same DNA. Micro-RNA and messenger RNA are very different. The discovery of messenger RNA got the Nobel Prize for Physiology in 2023. Messenger RNA carry the instructions for how to make proteins, while micro-RNA carry the management instructions of how much protein to make, if any. Micro-RNA are very important when organisms are developing and healing. Too much or too little micro-RNA change protein production in ways that can cause mutations, cancers, and disease. By understanding how micro-RNA regulate cells through blocking or promoting protein production, we may develop a better understanding of many diseases and how to treat them. There are currently several micro-RNA trials trying to figure out how to combat cardiovascular and kidney diseases. Victor Ambros is with the University of Massachusetts Chan Medical School, and Gary Ruvkun is at Massachusetts General Hospital and Harvard Medical School. Ambros and Ruvkun began making their discoveries in the 1990s after discovering anomalies in biologists' favourite roundworms, Caenorhabditis elegans. They noticed that if you disturbed a particular gene, these worms didn't develop properly. They couldn't figure out what was going on, so they went back to the lab. They learned that when the gene Lin4 was broken or not sufficiently active, it caused the same developmental defects seen as when the gene called Lin14 was too active. The two scientists showed that Lin4 was binding to messenger RNA that encoded Lin14, blocking it from being translated into protein in a never-before-seen mechanism for gene control. Their findings were published in 1993 in the journal Cell. Their research showed that tiny stretches of RNA known as microRNA could directly bind to messenger RNA, preventing the instructions for the corresponding protein from being read by the protein-making machinery in cells. 7 years later, Ruvkun's lab discovered the second microRNA, which helped convince other scientists that microRNAs were not simply an oddity of roundworms, but fundamental to all biology. Gene activity must be continually fine-tuned to adapt cellular functions to changing conditions in our bodies and in the environment. If gene regulation goes wrong, it can lead to serious diseases such as cancer, diabetes, or autoimmunity. This is a really important communication network between genes that enables the cells in our bodies to generate all kinds of different complex structures and functions. All the complexity you see in the plants and animals around you comes from the genes that we all contain, and most importantly, how those genes talk to each other. Neural network physics. The Physics Nobel Prize for 2024 went jointly to John Hopfield and Geoffrey Hinton for the invention of the neural network software that's the basis for most modern machine learning and artificial intelligence. Not only can you talk to neural networks, as well as creating and interpreting images, but they're invaluable for sorting and analysing huge amounts of data. This might sound like it's got more to do with mathematics than physics, but the first neural network software was developed by the insight that the physics of how atoms in a magnetic material affect each other's spin was remarkably similar to the way that nerve cells in a brain communicate to process information. It took a physicist studying brains to understand the similarity and make it work in software. In program software, the program receives data, which is processed according to a clear description and produces the results, much like when someone collects ingredients and processes them by following a recipe, producing a cake. Instead of this, in machine learning, the computer learns by example, which gives it the power to tackle problems that are too vague and complicated to be managed by step-by-step instructions. In 1949, Donald Hebbs published his hypothesis that learning occurs because connections between nerve cells, neurons, are reinforced when they work together. In 1982, physicist John Hopfield was studying the brain at Caltech and realised the similarity between the way the brain works and the way atoms in magnets work. Magnetic materials have special characteristics thanks to their atomic spin, a property that makes each atom a tiny magnet. The spins of neighbouring atoms affect each other, and this can allow domains to form with spin in the same direction. He was able to make a model network with nodes and connections by using the physics that describes how materials develop when the spins influence each other. The network that Hopfield built has nodes that are all joined together via connections of different strengths. Each node can store an individual value. In Hopfield's first network, this could be a 0 or a 1, like the pixels in a black and white picture. Hopfield described the overall state of the network with a property that's equivalent to the energy in the spin system found in physics. The energy is calculated using a formula that uses all the values of the nodes and all the strengths of the connections between them. The Hopfield network is programmed by an image being fed to the nodes, which are given the value of black 0 or white 1. The network's connections are then adjusted using the energy formula, so that the saved image gets low energy. When another pattern is fed into the network, there's a rule for going through the nodes one by one and checking whether the network has lower energy if the value of that node is changed. If it turns out that energy is reduced if a black pixel is white instead, then it changes colour. This procedure continues until it is impossible to find any further improvements to lower the energy of the system. When this point is reached, the network has often reproduced the original image on which it was trained. Hopfield's method is special because several pictures can be saved at the same time, and the network can usually tell the difference between them. Hopfield used the metaphor that searching the network for a saved state was like rolling a ball through a landscape of peaks and valleys, with friction that slows its movement. If the ball is dropped in a particular location, it will roll into the nearest valley and stop there. That's low energy. If the network is given a pattern that's close to one of the saved patterns, it will, in the same way, keep moving forward until it ends up at the bottom of a valley in the energy landscape, thus finding the closest pattern in its memory. The Hopfield network can be used to recreate data that contains noise or which has been partially erased. Hopfield improved the software so that the network included nodes that can store any value, not just 0 or 1. If you think again about nodes as pixels in a picture, they can have different colours, not just black or white. Improved methods have made it possible to save more pictures and to tell the difference between them even when they're very similar. The networks can identify or reconstruct any kind of information at all, provided it's built from many data points. Geoffrey Hinton read Hopfield's paper on his machine learning neural network and added processing based on the statistical physics of gases. It's impossible to track all the separate molecules in the gas, but it is possible to consider them collectively to determine the properties of the whole gas, such as pressure or temperature. There are many potential ways for gas molecules to spread through its volume at individual speeds and still result in the same collective properties. In the 19th century, Stefan Boltzmann wrote an equation to calculate the probability of each of these potential states, based on the energy available in the system. Geoffrey Hinton's improved network used Boltzmann's equation, and he published the method in 1985 as the Boltzmann machine. The Boltzmann machine worked with two different kinds of nodes. Information is fed to one group, which are the visible nodes. The other nodes form a hidden layer. The hidden nodes' values and connections contribute to the energy of the network as a whole. The machine is run by applying a rule for updating the values of the nodes one at a time. Eventually, the machine will enter a state in which the node's pattern can change, but the properties of the network as a whole remain the same. Each possible pattern will then have a specific probability that's determined by the network's energy according to Boltzmann's equation. When the machine finally stops, it has created a new pattern, which makes the Boltzmann machine an early example of a generative AI model. The Boltzmann machine can also learn, not from instructions, but by being given examples. It's trained by updating the values in the network's connections so that the example patterns, which were fed to the visible nodes when it was trained, have the highest chance of being output when the machine is run. The probability of this pattern being reproduced increases the more times the pattern is repeated in the training. Training also affects the chance of outputting new patterns that resemble the examples on which the machine was trained. A trained Boltzmann machine can recognize familiar traits in information it has never seen before, if the new information is in the same category as the training data. In 2006, Geoffrey Hinton and his colleagues developed a method for pre-training a network with a series of Boltzmann machines in layers, one on top of the other. In 1982, John Hopfield used a network with 30 nodes. If all the nodes are connected to each other, there are 435 connections. The nodes have their values, the connections have different strengths, and in total, there are fewer than 500 parameters or dimensions to keep track of. He also tried a network with 100 nodes, but this was too complicated for the computer that he had available. We can compare this to the large language models of today, which are built as networks that can contain more than 1 million parameters, or 1 trillion dimensions. Machine learning was used to sift through and process the vast amounts of data necessary to discover the Higgs particle. It's also been used to reduce noise in measurements of the gravitational waves from colliding black holes, in computational chemistry, and the search for exoplanets. Neural networks are changing the world.
You're listening to Ian Woolf on Diffusion Science Radio. Send emails to science.diffusionradio.com. We're brought to you across Australia on the Community Radio Network and podcast over the internet on diffusionradio.com. Oh, science and wonder, they go hand in hand, unlocking mysteries in this great big land. From galaxies and stars to the depths of the sea, science keeps revealing what's possible to be.
Computational chemistry. The 2024 Chemistry Nobel Prize was awarded jointly to Demis Hassabis and John M. Jumper for using machine learning to successfully predict the structure of the 200 million known forms of proteins, and to David Baker for using computational methods to create entirely new proteins that have never existed before. The proteins that form the chemistry of all life are generally built from 20 amino acids that can be combined in endless ways. Using the information stored in DNA as a blueprint, the amino acids are linked together in our cells to form long strings, which then twist and fold into a distinct, and sometimes unique, three-dimensional structure. This structure is what gives proteins their function, whether they go on to become part of muscles, bones, hair, or many other things. Many of them form enzymes, which drive life's chemical reactions with astounding precision and speed. The proteins that sit on the surfaces of cells are also important and function as communication channels between the cell and its surroundings. In 1962, the Nobel Prize in Chemistry went to the discovery of the three-dimensional shape of proteins using X-ray crystallography. In 1972, the Chemistry Nobel Prize went for the discovery that proteins always fold into the same shape depending on the amino acids they're made from. The paradox was that long chains of amino acids could randomly fold into an enormous number of three-dimensional shapes before ever reaching the one needed in the cell. But somehow, it always happens the same way in milliseconds in living cells. So it can't be random. The determination for the correct folding must be encoded somehow in the amino acid sequence. In 1994, a competition was started for people to try and predict the three-dimensional shape of a real protein that had been measured with X-ray crystallography, having been given only its amino acid sequence. The competition was called Critical Assessment of Protein Structure Prediction, CASP. The best CASP predictions were correct 40% of the time, but you needed over 90% to win. In 2010, chess master and neuroscience expert Demis Hassabis founded DeepMind, a company where he pioneered AI, and then sold the company to Google in 2014. He continued researching at DeepMind and registered for the CASP competition in 2018 with his new model AlphaFold. And he beat the best humans and achieved 60% accuracy in predicting the shape of a protein from its amino acid sequence. But of course this wasn't enough to win, you needed 90% or better accuracy. John M. Jumper joined DeepMind as a physicist who'd been using computers to simulate protein folding. He co-led the team with Hassabis, and together they developed AlphaFold 2, which used a new kind of neural network called transformers. Transformers can find patterns in enormous amounts of data in a more flexible manner than previously and efficiently determine what should be focused on to achieve a particular goal. Transformers are used in most AI systems in these last few years. The team trained AlphaFold 2 on the vast information in the databases of all known protein structures and amino acid sequences. They entered the 2020 CASP competition and got nearly 100% accuracy in predicting the shapes of the proteins from only knowing the amino acid sequences. They won. Google DeepMind has made the code for AlphaFold 2 publicly available and anyone can access it. By October 2024, AlphaFold 2 has been used by more than 2 million people from 190 countries. Previously, it often took years to obtain a protein structure with X-ray crystallography, if at all. Now it can be done in a few minutes. In 1998, David Baker developed his own computing system, Rosetta, and entered the CASP competition. He did well enough to wonder what would happen if the software could be run in reverse. If you could give it a protein function and have it predict which amino acid sequence would create that protein, even if it didn't exist anywhere before. This is known as de novo design. To do this, Rosetta searched a database of all known protein structures and looked for short fragments of proteins that had close to the desired structure. Using fundamental knowledge of protein's energy landscape, Rosetta then optimized these fragments and proposed an amino acid sequence. Baker's research group introduced the gene for the proposed amino acid sequence into bacteria. These bacteria produced the desired protein. Then they determined the protein's structure using X-ray crystallography. The protein that the researchers developed, TOP7, had almost exactly the structure they'd designed. TOP7's unique structure did not exist in nature, and with its 93 amino acids, the protein was larger than anything previously produced using de novo design. David Baker published his results in 2003, along with the code for the Rosetta software. After the 2020 CASP competition, where AlphaFold 2 won using transformers, David Baker added a transformer-based neural network to Rosetta, which has sped up the de novo design of proteins. It's incredible that we can now so easily visualize the structure of these small molecular machines. It allows us to better understand how life functions, including why some diseases develop, how antibiotic resistance occurs, and why some microbes can decompose plastic. The ability to create proteins that are loaded with new functions is just as amazing. This can lead to new nanomaterials, targeted drugs, more rapid development of vaccines, smaller sensors, and a greener chemical industry. The possibilities are endless.
In the lab where the wonders unfold, Victor Ambrose and Gary Rufkin, brave and bold, discovered micro-RNAs, a new way to see, regulating genes, shaping our destiny. From worms to humans, they paved the way, unlocking the secrets of life every day. Oh, Nobel Prize, shining so bright, for the secrets of life brought to life. From proteins to shapes and networks, they weave in the dance of the genes we believe. That came the minds of machine learning's grace. John Hopfield and Geoffrey Hinton took their place. Foundational discoveries in neural network song, enabling machines to learn and grow strong. With software inventions that changed the game, they open new doors to knowledge and fame. Oh, Nobel Prize, shining so bright, for the secrets of life brought to life. From proteins to shapes and networks, they weave in the dance of the genes we believe. Then came the minds of machine learning's grace. John Hopfield and Geoffrey Hinton took their place. Foundational discoveries in neural network song, enabling machines to learn and grow strong. With software inventions that changed the game, they open new doors to knowledge and fame. Oh, Nobel Prize, shining so bright, for the secrets of life brought to life. From proteins to shapes and networks, they weave in the dance of the genes we believe. David Baker crafted proteins anew with computational design that's cutting through. Demis Hassabis and John Jumper joined the fray, predicting structures in a revolutionary way. So here's to the winners who dare to explore, unlocking the mysteries at science's core. Ambrose and Rufkin for gene regulation's might, Hopfield and Hinton for machine learning's light. Oh, Nobel Prize, shining so bright, for the secrets of life brought to life. From proteins to shapes and networks, they weave in the dance of the genes we believe. Let's raise a glass to those who inspire, fueling our passion with scientific fire. The 2024 Nobel shine ever so clear, a celebration of knowledge we hold dear.
In the laboratories of Your Name Here. And here, dedicated scientists faced the challenge. Years of heartbreaking failures and setbacks only stiffened their resolve to conquer the problem.
That's it. That's the answer. We've done it. After all these years, we've invented it. How about that?
And with those historic words, the search was over. From the laboratories of Your Name Here had come the key to the secret that had baffled man through the ages. No longer a dream, but a reality was Your Product Here. Now, for the first time, limitless horizons opened for the nation. A brighter future unfolded. Thanks to Your Name Here. Employment boom. not only in the vast modern facilities of Your Name Here, but in factories everywhere, geared to supply this vital new industry that is reshaping our economy and transforming the lives of millions.
I produce Diffusion, which is broadcast around Australia to 28 stations on the Community Radio Network, including 2 MVR in Nambucca Valley, 3 MBR in the Mallee Border districts of Victoria and South Australia, City Park Radio 7 LTN in Launceston, Tasmania, 4 Triple D in Dalby in the Darling Downs in Queensland, 4 RPH in Brisbane, 6 GME Goolari in Broome, Western Australia, Lofty FM in the Adelaide Hills of South Australia, and 2XX FM in Canberra. Diffusion is narrowcast on Indigo FM 88 in northeast Victoria. And Diffusion is syndicated globally on astronomy.fm. Subscribe to the podcast on the Diffusion website, www.diffusionradio.com. That's www.diffusionradio.com, and check the website for links, photos and videos about this week's show. If you enjoyed this show, you can explore more than 1,000 previous episodes archived on diffusionradio.com, where the shows are labelled by keywords so you can focus in on the stories you want to hear. Join my patrons at patreon.com/diffusionradio. Make a donation through paypal.me/Ian Woolf. Support Diffusion by buying from our Etsy merch store, Diffusion Science. I'm Ian Woolf. Join us inside your audio device of choice for more science wondering next week on Diffusion Science Radio.
Science is fun. It helps you to learn, to know, and to appreciate. When you study science, you may go on field trips. You discover the marvelous interrelationships between all living things. You learn to read the history of the Earth as it is written in rocks and fossils. You find out what makes things tick, everything from a molecule to a living organism. In the study of science is found the most useful and satisfying knowledge of man. Knowledge of his physical world, its past, its present, and its future. And in your moments of relaxation, now and in the years to come, you will find the study of science leading you into fascinating pursuits. Photography, collecting. Why study science? Study science because you will find in the study of science a richer, more rewarding life.